Hi, good afternoon, everyone. So I just noticed that I'm the only one from Chulalongkorn University here. Are you guys from Chulalongkorn University? Raise your hand. Oh, okay. สวัสดีค่ะอาจารย์ค่ะ Today I'm going to talk about the topic. Maybe it's a specific topic because it's come from one of the paper that we recently got publication on the scientific reports. It's the Untargeted Metabolomics Review Pathway associated with neuroprotective effects of Oxyresveratrol in the neuroblastoma cells. Okay. ไม่ไปต้องกดเข้ามาตรงนี้ใช่ไหมคะด้านลง Okay. Here at Chulalongkorn University, about last year, I and my team just established the Metabolomics for Life Science Research Unit at Chulalongkorn University. Part of the faculty members from Faculty of Science and Faculty of Pharmaceutical Science. Okay, and for me, I'm from the Department of Food and Pharmaceutical Chemistry, the Faculty of Pharmaceutical Science, Chulalongkorn University. You probably think that at the Faculty of Pharmaceutical Science, we work only for the drug discovery and drug with the patient, but actually, we have the Department of Food Chemistry too, and there are only two university in Thailand that have this department, which are Chulalongkorn University and Mahidol University at the Faculty of Pharmaceutical Science. So the talk we're going to talk today is the work that my student, she just graduated with a PhD by this paper. She is Dr. Nurisan Mahamat. As you can see, she is a Muslim. So her research questions is quite different from others. She works at Halal Science Center at Chulalongkorn University, and then she was sent to study the PhD. So she came to me. So this is her with our previous Prime Minister, if you remember him. So she has the equipment in the science center, Halal Science Center, and she uses like, this is, she has the LCQ top, and she uses for like just a bit of work, and then she wanted to extend the work and the performance of this equipment. So she came to me, and I was just graduated at that time, and considered as an early career, and I wanted to develop the metabolomics as a new thing in my university. So I asked her, why don't we study on the metabolomics? But her research question, she wanted to develop things that serve the Muslim population and also the global population also. So what we look at that time is that we are currently in the aging society, which we already passed it in 2022. In Thailand, we currently in the aging society. So we wanted to find something that useful for the aging society, some product or some dietary supplements or some nutraceuticals that can be used for aging society for the protection of neurodegenerative disease. So this is what we think at the beginning. So from my department, so I wanted to clarify first what difference between the dietary supplement or what we call อาหารเสริม in Thailand versus drug. This is a different aspect. If you see here the graph, I really like this graph because here in the x-axis, this is the health status. This is time that this is a disease status or health status. So in the blue here, in the blue color here, this is metabolic stress, which means that you almost get the disease, but you not have the disease yet. You don't need to take the drug yet. Like when your lipid profile is high, but not yet taking the drug, maybe you can modify your lifestyle, modify the food. But once you pass this stage of metabolic stress, and then your lipid profile or the sugar profile is higher than the level, you need to take the drug. This is called metabolic syndrome. So for metabolic syndrome, it means you are a patient, you have the disease, you need something to cure you, so you need the drug here. So the drug is for therapy. But here, when you have the metabolic stress, you don't need to take any drug. But the thing is that you need to modify your lifestyle, and food is one thing that you can take here for the protection. So this is the role of food versus the role of the drug, which are different. So when you do the experimental design, it's got to be different too. So for the drugs here, it's about the patient and disease. It's about the efficacy that you want it to be here versus the toxicity if you take more drug, too much of drug. But for the consumer and health, this is the food or the supplement, which is registration as a food. So it goes from absorption, distribution, metabolism, and excretion too. But the thing that we wanted here is for the role of the prevention, not for the therapy. So here is for the prevention, not for the treatment for the main role. So this is the thing. So it comes out with the idea about the metabolomics. What is the role of the metabolomics technology in the dietary supplements development? So this is again metabolomics. Maybe some speaker already mentioned it at the beginning, but maybe I mentioned it again. Maybe it sounds cliche, but for better understanding, metabolomic is the high-throughput screening of the complete set of the metabolite in a biological sample. And that biological sample can be cells, can be body fluids, or can be tissue. And this is the whole metabolome that you may see in the morning from the professor from Singapore here. So we wanted to identify all at once. So actually, the metabolite identification is not new. If we identify or quantify only a few metabolites, like your blood sugar, something like that, it's not new because everyone had a health checkup every year, right, for the sugar. This is metabolite. But if you identify the endogenous metabolite all at one time, we call it metabolomics. So this is a step in metabolomics analysis. So for the metabolomics sample, it can be, if it's in a clinical study, it can be biofluids like serum or urine. In our lab, we use the breath sample to exhale breath sample. It can be anything from the biological organisms. It can be cells. What we're going to talk about here today is about the cell, because the cell is less complex. We can control everything. So if you wanted to see the underlying mechanism, cell line could be one thing that you consider at the beginning. So it can be tissue too. If you can have the tissue from biopsy from the human subject or maybe the animal, so you can use it too after you test with the bioactive compound that you're interested, and then you collect the sample, and then go to the separation part here for the chromatography and mass spectrometry for the detection, and then you have the chromatogram like this. And then after you process the data, you do the data visualization and you see things like this. You can identify the difference between the metabolite profile between the two sample sets or more than two. Or you can use the profile and go for the pathway mapping or pathway analysis, and you can find that which pathway that bioactive compound that you interest will affect you. So in terms of food or nutrition, what can metabolomics do? It can be used to find the relationship between food and health. It can be used to find the underlying mechanism of the food. I mean, if you are the patient, you have the disease, you already have the marker already. Like if you have like diabetes, for example, you have high blood sugar level, and you know that that sugar is the biomarker. But if you wanted to test some efficacy of your bioactive compound, how can you test that? Is the thing that is a little bit changes over time, because for the dietary supplement, the one who take it, it's a healthy subject, right? It's not the patient, mainly the healthy subject. How can you see that little changes? So we need the technology to find something like this, to find a small thing like this. And this is the challenge for the work in the food and dietary supplement study. So we can use metabolomics for the biomarker study. We can find the biomarker relevant to the food consumption that we give to the subject, and better insight into the genetic polymorphism and the nutrient metabolism, and select of the good bioactive ingredient, which one make the good, make subject have a better health, or in the future, maybe we can put it for the functional claims. So then we come up with this idea. We wanted to find some kind of ingredient for aging society and use metabolomics as a platform to see the efficacy of that ingredient. So when we say the aging society, the things that come to everyone is about the neurodegenerative diseases such as the dementia, Alzheimer, or Parkinson's. This kind of disease like Alzheimer and Parkinson's, right now, currently, nowadays, no drugs that can cure, 100% cure for the Parkinson or Alzheimer diseases. The only thing is that the drug can slow down the progress of the disease, but not 100% cured. So is it better if we can find some kind of ingredient that can slow down this disease? Because at the end, everyone got to be aged. Everyone gonna be old. But if you can find some ingredient, it would be great. Aging society, aging metabolism is directly related to the oxidative stress in the body, which is related to the neuronal cell death, and is about the neurodegeneration. So at the beginning, we think about the resveratrol. Why? Because the resveratrol has been proved that it has the antioxidant activity, it's cardioprotective, it anti-cancer, it has estrogenic activity, it has anti-inflammatory effect, it has neuroprotective that we want, it has anti-diabetes, and it prolongs lifespan. And this is the only compound that nowadays have the prolonged lifespan effect. But the problem is that the resveratrol mainly found from wine. And Muslims cannot drink alcohol. This is the problem of the product derived from alcohol. So we cannot use resveratrol. So the Halal Science Center wanted to find another ingredient that can be replaced the resveratrol and can be served for the Muslim population. So we come up with this compound. It's called Oxyresveratrol, which can be found from many plants and also from the bark of the lacucha plant. This is no alcohol. And its structure is just modified from the resveratrol, which make it more water soluble with the more hydroxyl groups in that. And actually, it has been proved in the cells that it has the antioxidant activity, anti-inflammatory, and the neuroprotective effects. It's already proven in the cell, but why we do this? Because we don't know the underlying mechanism yet, why it's neuroprotective, which metabolism that it affects, which endogenous metabolite that it change after taking this kind of compound. So the objective of the study is to investigate the potential neuroprotective mechanism of the oxyresveratrol by metabolomics. platform, and elucidate the mechanisms and investigate the neuroprotective effects of the oxyresveratrol in the neuroblastoma cell line, and to identify that, to find that if the neuroprotective marker can be useful to use in the future. So we have the things to we have the experiment to confirm that. So for our experiment, we use the cell line. If we want to find the underlying mechanism of something, we want the sample that it can be controlled. So we selected the cells rather than the animal model or the human subject. So we divided the cell into 4 groups. The first one is the control, nothing, and the rotenone toxicity. It's the rotenone-induced toxicity in the cells, and the oxyresveratrol with the rotenone and the oxyresveratrol alone. The important thing is that, remember, if you wanted to test for the food supplement, it's different from the drug. The drug, you induce the cell to have the disease first. For example, induce it by the rotenones first, so the cell is damaged, and then you give it with the drugs. But for the dietary supplement or food supplement that we mean to use in healthy subject, we got to give the oxyresveratrol first to the cell, just like everyone taking the supplement first before you get the disease like protective effect, and then after that we induce it with the rotenone later on. So we divided the experimental design into 2 main parts, which is the cellular assays and the metabolomics analysis. And for the cellular assay, we see we carry out the experiment about cell viability test, the antioxidant analysis, and the detection of the protein expression. And for the metabolomics analysis, we see the metabolite profile and the candidate marker and the related pathway. First, for the cellular assays. For the cellular assay, so first we find the optimum dose of the oxyresveratrol against the rotenone toxicity. So we find the suitable dose of the oxyresveratrol and then the rotenone and the effect of the oxyresveratrol and rotenone together to the cells. And after that, we confirm the antioxidant property of the oxyresveratrol in this experiment. So as you can see, the red one is the control, and then the pink one is the is the rotenone-induced toxicity test, and the blue one, it's oxyresveratrol in 2 doses, 10 and 20 micromolar. So here is the mitochondrial membrane potential, and here is the reactive oxygen species, which is going to be opposite to the glutathione level. If the reactive oxygen species raise high, which means the toxicity occurs, and after if we have the oxyresveratrol first and then induced with the rotenone, you can see that the levels of the RS goes down. The same thing for the glutathione. It's the opposite. It means that if you get the toxicity by the rotenone, the level of the glutathione goes down, and then after you, if you receive the oxyresveratrol, it goes up a little bit, but still lower compared to the control. And next, we find the most suitable dose for the neuroprotective effect of the oxyresveratrol, because we wanted to select only 1 dose for the metabolomics analysis. So we carry out the experiment to see the protein content, and then we find that at the 20 micromolar, it gives a better result. So we select that dose for metabolomics analysis. So for the metabolomics analysis in the cell line, we begin with the cell, which is, for the cell, it should have been to about 10 to the minus 6 cells per well, and you should have at least 8 flags separated, not just like 1 and then you separate it to 8. You should raise like 8 8 plates separately, and then extract the metabolite. And then we did the data acquisition by LCQ-TOF MSMS, and did the data processing by MS-DIAL, and then statistical analysis and pathway mapping by the metaboanalyst. So for the extracting metabolite, so we grow the cells in here, 8 per group, and then we use the scrape method to take the cells, and we wanted the intracellular metabolite extraction. After that, we have the cell extraction here. For the metabolomics analysis, we use liquid chromatography mass spectrometry, which is the quadruple time of light, which considers the high resolution technique. So here is the equipment at the Hala Research Center, and we thank Dr. Jaran here from Brugge. He is a specialist. He helps us a lot for running this equipment. Here is the LCQ-TOF. This is the condition that we use. And after that, we process the data because there are going to be a lot of metabolite that come out at one time. So we use the MS-DIAL for the data processing. So for the result, we match the with the 4 database, which is authentic, standard, GNPS, mass bank, and RESPECT. And then from the about 6,000 metabolites that come out from the LCQ-TOF, we end up with about 294 metabolites by data processing step. And here is a metabolite profiling. So this is a group. This is a PLS, partial least square discriminant analysis test. This is a QC sample, which is very needed if you run the metabolomics to check the equipment performance. And as you can see that if the cell has the retinol toxicity, it will chip far away from the control. And if it has a oxyresveratrol before the retinol, it kind of shift back a little. But still it's not as obvious. So we go for the VIP score, which is a variable important in protection here. So if the value is high, it means it's a good marker to separate between the group. So we can see, as you can see, that is only one, that is one metabolite showing the VIP score higher than 3. So normally, if it's higher than 1.5 or 2, it's high. But here is very high. It's more than 3. And this is the 7,8 dihydrobiopterin. So we're very interested into this metabolite because this is not normal. Normal is in the rate or control. When you have the retinol toxicity in the cell, it goes down here, like this. But if you have the oxyresveratrol before you expose to the retinol, the level is not going down. So there should be something about this kind of compound. So we try to research this compound and the related pathway. So this is the raw data sets that we have. We match each compound that we have with the HMDB database. It's the human metabolome database, which is like Human Genome Project, but this is a human metabolome database. So you can check it in the website. It's free. And then if you put the numbers, the name in there, or the entry key, or the cache number, you will have the HMDB ID. And this ID will go for the pathway mapping in the metabolomics software. And then you will see the pathway that impact to that the oxyresveratrol impact. And here we see that the phenylalanine, tyrosine, and tryptophan metabolism is the most related pathway. And also is the enrichment ratio is the highest one. So this is the pathway that we are interested in. So we hypothesize that Oxyresveratrol should have done something that related to the 7A dihydrobiopterin. And this compound we researched it, and it's related to the dopamine metabolism. So we wanted to check it, so we carry out another cell line experiment. We did another set of the experiment, not that one that we did, the untargeted metabolomics. We have another set of the sample to check if our target or our biomarker that we found is correct or not, is it can be used in the future. So we collect another experiment, which is very important. If you carry out the clinical trial for some drug or for something, you should have another set of the subject. This is also the same thing. So after we carry out another experiment, and we extracted the chromatogram of our biomarker that we interested in 7A dihydrobiopterin, and we found that the level in the picture is here, and then this is the number in the table. And the intensity of the peak area is significantly different, and it went the same way as an untargeted metabolomic does. So for the control, for the control is high, the level is high, and for the rotinone is significantly lower than the control. But if you are exposed to, you got oxyresveratrol first, treatment first before exposed to the rotinone, the level goes up. So for the untargeted metabolomic, the things that we're gonna get is the hypothesis generating, which is like kind of like a flip opposite to another normal experiment, because the normal experiment you make the hypothesis first, and then after that you get the results. But for metabolomic, it's the thing that you found new thing and then you generate a new hypothesis. So we hypothesized about the underlying mechanism of the neuroprotective effect of the oxyresveratrol like this. So this is our biomarker, 7A dihydrobiopterin. It serves as a cofactor of the enzyme phenylalanine hydroxylase and tyrosine hydroxylase. So it converts phenylalanine to tyrosine, and then it helps, it's a cofactor of the enzyme tyrosine hydroxylase, converting tyrosine to dopa. And then dopa is a substrate for the dopamine. And why the dopamine is very important? Because this is pathophysiology of the Parkinson's disease in the brain. So if you are healthy, you will have a lot of dopamine levels in your body. But if you have the Parkinson's disease, your dopamine level will go down. So here, if your substrate is high, you will produce more dopamine. And this is the underlying mechanism that we propose, and how oxyresveratrol can be used for the neuroprotective effects. So in conclusion, first we did the cellular assay, and the cellular assay confirmed that oxyresveratrol was effectively prevent rotenone-induced cell death and the decreased reactive oxygen species level or RS level in the cells. And after that, we did the untargeted metabolomics analysis to find the metabolite profile, also the biomarker, and we found that the 7A dihydrobiopterin can be served as the key metabolite or the biomarker of this study. And then we researched that, and we did pathway analysis and found that the 7A dihydrobiopterin was the biomarker that related to the phenylalanine, tyrosine, and tryptophan metabolism, which is served as the cofactor for the function of the enzyme in the dopamine biosynthesis pathway, which is very important for the neurodegenerative disease. And after that, we did targeted analysis to confirm the marker that we found, and we found that this 7A dihydrobiopterin is the effective biomarker. So we did the experiment again, and the result turned to be the same way. So this information is useful if we can use this cofactor as the indicator for treatment, no, for prevention, for the oxyresveratrol, for using oxyresveratrol as a prevention for the neurodegenerative disease in the future in the aging society, and can be the future guides for this research area to develop the novel therapeutic approach or the preventive approach for the neurodegenerative disease in the aging society. So, Will, thank you very much and hope that this information can be useful for you to carry out the new experiment in the future. Thank you.
ขอบพระคุณผู้ช่วยศาสตราจารย์เภสัชกรหญิงดร.โรสริน ตันสวัสดิ์เป็นอย่างสูงค่ะ
Is there any question from the floor? Yes, please.
Thank you for this nice talk. It's not a question. I like to make a comment. I think, the wonderful work. This is a typical work that you use untargeted analysis and then find the biomarker, and then you went on to do validation. Not many people do that. So I really want to say, appreciate this. It's really very good work. Thank you very much.
Thank you very much. We aim to find the neurodegenerative marker for this study.
Yeah, it's quite interesting about the anti-aging compound and the rejuvenated stuff. So my question is that because it's an oxyresveratrol is a natural compound, right? So I'm not sure about the stability in our body. Like if we consume it, how long it's gonna be last in our body?
Thank you very much for the questions. Actually, I searched for that. It's not registration to eat yet, but somebody uses as a cream. It still need for the FDA registration in Thailand some more experiments for to prove that it is good enough.
Thank you very much.
Thank you for very informative, very solid methodology, especially for the validation step. But could you please explain me why you use the neuroblastoma cells line for the representative for neurodegenerative?
Okay, thank you. Okay, that's a good question. I confess that I'm not a cell person. We have collaboration from Mahidol University, but for the neurodegenerative disease, we use neuroblastoma cell, which is not the normal cell that we normally in the cell experiment we should use the cancer cell so it grows fast, so we can see what's going on. But for in the detail, I'm sorry, I cannot go for the deep answer for that, but you can have my contact and I can give you the answer later on and email you something like that.
Yeah, thank you very much.
You.
This gentleman, please.
Thank you for your nice presentation. Do you think somehow this new pathway involved with a gut microbiome? And have you considered do experiment on gut microbiome intervention to see whether gut change on oxyresveratrol?
Thank you very much. This is a very interesting topic and the aspect. If you wanted to carry out the experiment in the future, maybe you can help me to do the gut microbiome things, and we can do together with the metabolite profiling that our expertise. That's very interesting. Yeah, collect the stool and the blood. Yes.